Ao vivo.
Ok. Então, boa tarde, pessoal. Obrigada por estarem aqui hoje. Vou começar a falar inglês um pouquinho, tá? Para introduzir aqui a Akiko. So we are delighted to have Akiko Iwasaki at our SBI.ImunoTalks today. Akiko is a professor at the Yale University, USA, and a PI at the Howard Hughes Medical Institute, and a member of the National Academy of Sciences, USA. Akiko is an immunologist with multiple interests, especially regarding immune response to viruses. And during these challenging days, she is contributing a lot to the understanding of the immune response against SARS-CoV-2. Besides being a brilliant scientist, Akiko is a mom of two lucky girls, Naomi and Emi, and an active and essential voice supporting gender equity and diversity in science. And with all, we are very thankful, Akiko, for accepting our invitation. Thank you again.
Thank you very much, Karina, for the nice introduction. I'm delighted to be here, and I will present our recent work on immunology to SARS-CoV-2. Share this. Okay. Can you see? It's okay? Okay, good. So yes, I'm delighted to be here today to share about our recent work on immune response to SARS-CoV-2. And I'll start with a little introduction to both the virus and the immune system and what we know and what we don't know. SARS-CoV-2, as you all know, is a member of the coronavirus family. It consists of a large genome of a positive sense RNA, about 30 kilobases in size. It is enveloped and it has these spike protein that protrudes from the envelope, and that becomes the vehicle by which the virus uses to enter host cells, the spike protein, as well as target of antibodies and T cell responses. And we are not completely naive to coronaviruses in general. There are many other sort of seasonal coronaviruses that infect humans, and it's thought to cause 30% of all common colds, including these alpha and beta coronaviruses. But there are these zoonotic coronaviruses that cause severe to lethal infections, including the SARS-CoV-1 and MERS-CoV. And of course, joining this rank is the SARS-CoV-2. So SARS-CoV-2, like any other viruses, utilizes host receptor for entry. And in this case, it's the human ACE2. And this allows the virus to enter the cell, fuse, and inject its genomic RNA. That can be replicated to make more copies, as well as produce virus to assemble its component and bud out of the membrane to infect other cells. So with respect to this virus, how does it compare to other respiratory infections? And influenza virus is another formidable viral infection that causes disease and lethality in humans. But if you look at the R0 value, which is the basic reproductive number of an infected person capable of transmitting to other individuals, you see that the COVID-19, the SARS-CoV-2, has much higher levels of R0 currently, because there is no pre-existing immunity in humans against this, at least no existing pre-existing antibody response to COVID-19. And the case fatality rate obviously depends on the demographics as well as geographic locations, but it's estimated to be a lot higher, especially in older individuals. And the other thing that makes this virus quite successful is it has a very long incubation period between the time that you contract this virus to the time you become symptomatic, and that can last for up to 14 days, in which during which time the virus replication is can reach quite high levels to transmit to others. And of course, the other kinds of zoonotic coronaviruses also have high R0 value, as well as quite high lethality in the case of MERS virus. So what can history and virology tell us about how we might deal with this virus? We know that antibodies to common cold coronaviruses don't last for very long, and the immunity seems to wane within a year, so that people become reinfected with the same virus during season to season. And people who have recovered from the common cold virus may become reinfected, but they're mostly protected from disease. And we have evidence of reinfection now with the SARS-CoV-2 coming out of multiple countries. This list keeps growing every day. I can't keep up, but there are many other countries added to this list, including India and other places. And so we know that the reinfection can happen. How frequent it is and what the consequences are still murky, since there are so few cases right now, and we don't know the immune status of people who are reinfected. It's very difficult to draw conclusions at this point and what controls reinfection. And of course, the only safe way to deal with this infection is through vaccination. Even though we as in humans have not produced any vaccines against coronaviruses yet, there are over 180 different vaccines being currently tested in various stages. Some of them are quite far along in phase 3 clinical trial. Another thing to mention about the respiratory viruses is that many of them have seasonality, including the influenza virus, as well as the other human coronaviruses that cause the common cold. And we have been interested in what the impact of these environmental factors might have on the immune resistance against respiratory infections. And so far, we know that the lower temperature can impair innate resistance against viruses because of the impaired Brigg-like receptor signaling pathway at temperatures that are found in the nose during the winter time. We also know from more recent work that absolute humidity really makes an impact on the host resistance to virus. And what we understand is that the lower humidity that's found indoors during the winter months, especially where I live in Connecticut, we tend to get pretty cold winter, and we basically, by raising the temperature of the air that we import from the outside, which already has low humidity, we make the relative humidity quite low during the winter months, and that impacts the ability of the mucociliary clearance of the virus from the upper airways, as well as resistance. Interferon responses are impaired by this kind of environment. So we are learning more and more about what the seasonality and what features of seasonality impacts the immune resistance to the virus. And of course, not to mention that these factors also impact the virus itself, its ability to remain airborne and to be stable. So I do worry about going into the winter season without having a vaccine yet. So what kind of research needs to be done? Well, today I'm going to focus mostly on immune analysis of this coronavirus, but we really need to, going forward, develop better antiviral agents. Investment in this area is very important for any kind of future pandemics. We don't know what kind of viruses or bacteria might cause future pandemic, and we need to be ready and prepared with medicine against those, as well as great, safe, and effective monoclonal antibodies that can be developed quickly against these viruses. You've probably heard a lot about different trials that are ongoing with Regeneron, monoclonal antibody cocktails that was used for the President of the United States recently. And of course, many other drugs and biologics to promote disease tolerance, which is really allowing the host to tolerate the impact of these types of infections. and of course, vaccine strategies that can be quickly co-opted for any kind of viral infection would be important. So one thing that's coming out of the literature in the last 9 months or so about COVID-19 is that it's very heterogeneous. It causes very different symptoms and manifestations depending on the host. So anywhere from completely asymptomatic infection all the way to severe disease and death. And also we see acute disease as well as long hauler disease, which people are suffering for months and months from variety of symptoms after recovery from this infection. And of course, it really also depends on the host demographics, the age as well as sex, and of course, pregnancy status. And so today I'm going to focus on a couple of different issues. One is to compare moderate and severe disease immune response to this coronavirus. And another point that I want to make is the comparison between male and female sexes with respect to immune response to this virus. And, one of the features that are coming out, which I won't have time to talk about today, is sort of the neurologic symptoms that occur after infection with this virus. And we have a preprint demonstrating that there is actually evidence of infection of neurons within the cortex of a deceased patient from coronavirus. And now there are more evidence coming out that not only does it cause inflammation in the central nervous system, but it might also cause direct infection in some cases. So I'm just going to give one introductory immunology slide, which is this, just to remind everyone that there are different types of immune responses that are dedicated to defending against different types of pathogens. And that is dictated by the interaction between naive T cells and dendritic cells. And when dendritic cells are stimulated through different types of pattern recognition receptors, they they secrete first order cytokines that allow T cells to become differentiated into different types of CD4 T cell subsets, follicular helper cells that assist B cell response, Th2 cells that are particularly suited for anti-helminths infection, Th1 cells for antiviral and antibacterial, as well as Th17 for antifungal and excellular bacteria. And of course, the regulatory cells that dampen exuberant immune response. So what I'm going to show you today is that in COVID-19, in the severe cases, all of these types of immune responses are engaged in the patients. So first, I want to talk about the mouse model of COVID-19. This is a good primer going into the human studies because we demonstrate an interesting feature of this virus with respect to the relationship to interferons. So as I told you earlier, the human ACE2 is the receptor for this virus. And mouse ACE2 is not capable of allowing infection with this virus. So we came up with a strategy to make a mouse model of COVID-19, in which we first introduced human ACE2 into the respiratory tract using AAV vector. And 2 weeks later, we can come back and then infect the animals with SARS-CoV-2. And what this is allow us to study replicating virus, both measured by RNA levels as well as plaque assays, and showing that this transduction with human ACE2 allows us to infect the animals and study replication within the lung. And not only that, these mice develop very good immune response, adaptive immunity. As shown here, antibody to spike-1, IgG, can be measured very quickly within 7 days. We have almost half maximum titer of the antibody generated. And within 2 weeks, we have a very high level of antibody generated in these mice. So we can study innate and adaptive immune response using this strategy. and where in the infection in the lung is the infection occurring. So when we stain for SARS-CoV-2 nucleocapsid antigen here, we see that there are cells that are infected. It appears to be cells within the alveoli. This is a staining for epithelial cells, the EpCAM staining in the green. You see these large conducting airways, but they're not the ones that are infected with the virus, but rather these sort of smaller alveolar cells that appear to be infected with this virus. And we can also look at pathological impact of this infection. You see a robust recruitment of leukocytes in the animals that have been transduced and then infected with SARS-CoV-2. So this is a very versatile animal model in which we can basically take any genetic knockout mice transgenics, strains of mice to transduce human ACE2 and allow them to become infectable hosts. And so one of the first things we examined is to see how important is the type 1 interferon response in controlling this virus. And so this is a review that we wrote about different innate sensors that are triggered as a result of this infection, leading to both inflammatory as well as type 1, type 3 interferon response. And once the interferons are generated, it will bind to neighboring cells and induce lots of interferon-stimulated genes that control the viral replication. However, this virus, as well as many coronaviruses, have devised methods to interfere with every step along the way of recognition and signaling. Even interferon type 1 receptor signaling can be blocked by viral encoded proteins. So we wanted to understand what is the relative importance of induction of interferon. So we can do that by using animals that are lacking the key transcription factors needed for transcribing interferon genes, as well as mice that are deficient in the interferon receptor. This allows us to look at the impact of interferon signaling for antiviral control. And just to show you that indeed animals that are transduced with ACE2 and infected with the SARS-CoV-2 induce lots of interferon-stimulated genes, and many of them are inducible by type 1 interferons. And this is also consistent with human respiratory transcriptome that was published, that 73% of the ISGs are shared between our animal model to the human respiratory tract. And but when we did this experiment in which we used interferon deficient animals or IRF37 deficient animals, and then we infected these animals with SARS-CoV-2 after ACE2 transduction, what we see is very little impact of the deficiency of interferon receptor or interferon transcription factors. So this indicated to us that even though interferon is engaging and inducing ISGs to some extent, they're really not very good at controlling the virus. However, what the interferons appear to be doing is that they're allowing the recruitment of leukocytes, such as inflammatory monocytes, monocyte-derived macrophages, as well as T cells and NK cells into the lung, and possibly contributing to disease pathogenesis. So what this told us is that, we did develop this very adaptable model of COVID-19 using ACE2 transduction by AAV, and lack of interferon or interferon induction appeared to have very little impact on viral replication. However, it did have impact on recruitment of leukocytes into the lung. And so if you were to imagine two different scenarios after an infection, this is a sort of scenario that's ideal where you have a viral replication starting to happen, which is accompanied immediately by the interferon response that's controlling the virus load and then recovering with mild disease. Whereas what we are seeing here appear to coincide with this model in which the viral load is quickly replicating. And the interferon response, even though it's induced, it may be too little or too late to control the viral persistence. And it leads to this severe disease phenotype. So armed with this finding, we went in to look at what is going on in the human immune response to SARS-CoV-2. So this human study was enabled by the fact that Dr. Albert Koh, who is a professor of School of Public Health at Yale, began this repository that allows us to collect samples over time from patients and study them in real time. So what this impact study, what we're calling this impact by repository, allowed us to do is to enroll patients in the hospital and then collect a variety of biospecimens from those patients every 3 to 4 days, allowing us to do longitudinal studies. And then we also have healthcare workers who are not infected with coronavirus, but served as a wonderful sort of control group from the same location. And as well, we were assaying for their viral load very rigorously, allowing us to pick up infected cases as soon as possible to alert the person who are infected, as well as to kind of allow quarantine of that or isolation of that person so that the healthcare worker will not transmit to other people. And then we also have the outpatient cohort. We're just starting to recruit people to study what happens in milder cases. But today I'm going to talk about the inpatient cohort. And so we started collecting PBMC from patients that are admitted to our hospital, and then analyze their leukocyte, both lymphocytes and leukocyte proportions, as well as activation status and cytokines. And so this longitudinal study it consisted of 3 groups. One is the uninfected healthcare workers, and then the moderate patients, moderate disease patients, 80 patients were recruited, and severe patients, 33 patients were recruited. And we also designated clinical scores to indicate the severity of disease all the way to death and mechanical ventilation, as well as supplemental oxygen levels that were required to treat the patients at any given time. So the first thing we noticed was, as reported by many others, we see a drop in the number of T cells in COVID patients. So the green is the healthcare workers, and you see that this is sort of the normal level of T cells that you find in the blood, whereas the moderate and severe patients have a drop in the T cell count in their blood. But inversely, we see increase in monocytes, neutrophils, and eosinophils that are elevated in severe and moderate patients. And we also see lots of cytokines, so-called the cytokine storm cytokines that are elevated in patients. In particular, you'll see the IL-6 is quite highly elevated in severe cases, IL-18, IL-1, and so on, including type 1 interferons that are elevated, as well as chemokines and this Th17 cytokines and so on. And one big clue as to why patients were moderate versus severe came from this comparison of longitudinal analysis of the viral load in the nasopharyngeal swab. And what we saw was that in the severe patients, they were unable to control the virus and maintain high levels of virus throughout the course of study, whereas the moderate patients were able to eventually control the viral load and recover from this infection. And if you look at the key antiviral cytokines, such as interferon alpha and interferon gamma, what you see is that there's a correlation between the viral load and the amount of these cytokines, indicating that it's the viral load that's driving these interferon responses, and that higher levels of interferon is not leading to reduction in viral load. So again, this is kind of reminiscent of what we saw in the mouse model. And similar things can be seen with TNF and TRAIL. And if you look at the first 12 days since the symptom onset in patients who either died in pink or discharged from the hospital in blue, what we see is that there is an elevated level of interferon alpha in the patients that ended up dying of this disease compared to those that recovered. And similarly, IL-1 receptor antagonist is also elevated in the diseased patients. And we also saw similar trends in other cytokines, indicating that, again, elevated levels of interferon seem to correlate with mortality as opposed to recovery in this case. So if you look across the board on different cell types and cytokines that we measured, what we see is this sort of unusual feature in which severe patients have increased levels of these inflammatory cell types as well as cytokines throughout the course of disease. Whereas in moderate patients, either they have a flat line or a reduction in the levels of these cytokines, regardless of the kinds of cytokines that we were looking at, including the type 1 cytokines, type 2 cytokines that are sort of thought to be most appropriate for parasite response, type 3 response, such as antifungal type of cells and cytokines are also being elevated in the severe patients. And something as unusual as IgE becomes elevated and maintained in the severe patients. Eosinophils were also another cell type that we saw in the severe patients to be elevated. So if you take all these cytokines and ask if there's any pattern of clustering of the patients based on those cytokines, what we see is that the unsupervised clustering analysis reveals 3 different clusters. The first cluster of patients have the highest levels of cytokines belonging to the tissue repair growth factor family members, such as PDGF, EGF, VEGF, as well as IL-7, so the darker the color, the more secretion of these cytokine we find in their blood. The second cluster of patients, in addition to this tissue repair factor, we see enrichment of proteins belonging to chemokines and mixed cytokines that are type 1, 2, 3. And then in cluster 3, we see the highest level of enrichment in the type 2 and 3 cytokine, as well as mixed cytokines. So if you look at the different age group of these different clusters, we see not a huge difference between the age of the cluster 1, 2, and 3, and length of hospital stay appears to be slightly higher in the cluster 3. But what's striking is that the trajectory of disease differ dramatically between the first cluster and the second and third. So people in the first cluster, which had the enrichment in the tissue repair growth factors, are able to maintain a low disease score and eventually recover from infection. Whereas many of the people in the second and the third cluster went on to develop significant disease, and unfortunately, some of these patients also died of this infection. And if you look at all the different factors and cell types that we measured and ask which ones are biomarkers for mortality, what we see is that the number one factor that is highly associated with mortality is IL-18, which, as you know, is an inflammasome-dependent cytokine. And interferon alpha is number 2, and then IL-10 and many other factors you can see here along the spectrum, indicating that the severe mortality-related factors are really kind of belong to this inflammasome factors as well as perhaps type 1 interferon. So this longitudinal analysis allowed us to really kind of segregate the different responses that are occurring in moderate and severe patients. And one of the biggest factors we believe that are driving this disease difference is really the viral load. And viral load is what's driving the cytokine responses. And those cytokines that are increased within 12 days of symptom onset is higher in the people who died of this infection. And we also identify different signatures of cytokine that can predict disease trajectory. So again, if I use the same graph, it appears that the second scenario is what's going on in the severe cases, that we have this elevated level of interferon kind of leading to inflammation and severity, potentially leading to inflammation and severity. So in the last part of my talk, I want to discuss using the same sets of patients, if there's any sex differences in immune response. And this question was raised because there are multiple reports coming from different areas of the world indicating that male sex is a risk factor for severe COVID-19 as well as mortality. And these are just some of the examples of what we see with respect to men accounting for higher levels of death and severe disease as well as hospitalization. And COVID-19 is not the only disease in which this sex disparity has been seen. Many other infections are known to have both the difference in the prevalence or incidence as well as severity of disease from infection with HIV, influenza, MERS, hepatitis, and so on. And so COVID-19 appears to also belong in this category of male severity and male mortality being more prevalent. So why is that? Well, we decided to tackle this question by looking at the baseline immune response in the patients that are coming into our hospital. And we excluded patients who are in the ICU or who are already being treated with tocilizumab, which is an anti-IL-6 receptor antibody, or on high-dose corticosteroids, because we wanted to focus on the naturally occurring immune response to this virus. And obviously these anti-inflammatory agents will interfere with that. So we just focus on the first sample from people who are moderate disease, who were not on these anti-inflammatory medicines. And so this is the cohort A, which consisted of 39 patients, 17 men and 22 women. And we also recruited healthcare workers. And then also we extended the analysis to a larger cohort in the end to see whether the findings hold in all stages of disease. So looking at this baseline, what we saw was that there was no significant difference between the viral load from the NP swab or the saliva or the antibody response to the spike protein here. So the male patients are indicated in orange and female patients are indicated in purple. And so you can see there is no statistically significant difference between these levels. And there are also chemokines and cytokines that we saw were elevated in both sexes of patients who were infected with COVID. And here you can see, for instance, CCL5 is elevated in both sexes and CXCL10 is also elevated. CXCL10 appears to be the most differentially elevated chemokine that we can measure. This is a really high level that we can measure in the blood of these patients, even early on. However, there were some factors that were differentially upregulated in male versus female. So in male patients, what we see is an elevated IL-8 and IL-18. As I mentioned, IL-18 is one of the biomarker, the top biomarker for mortality, and we see that elevated in male patients compared to the female patients. And if you look at the different cell types, we also see that the non-classical monocytes appear to be elevated in male compared to the female patients. And what's interesting is if you plot the level of CCL5 and these inflammatory monocytes, we see that there's a linear relationship only in the male patients between the CCL5 levels and the non-classical monocytes, whereas there was no such correlation in the female. Conversely, we see elevated activation of T cells, CD4s and CD8s, in the female patients compared to the male patients. So we wondered if these baseline data can tell us anything about the disease trajectory of these patients. So what we did was we took that group and then divided them into those that went on to worse disease, which are the filled circles, and those that had stable disease, which are the empty circles. And again, the males are in orange and females are in purple. You see that the age is elevated in male patients who had worse disease outcome, whereas there was no difference in female patients. And similarly, the body mass index is higher in the male patients who went on to develop worse disease compared to those who had stable disease. And there was no significant difference in the female patients. And so I told you that male patients tended to have less T cell activation. But if you look at the those patients that had very little T cell activation, this is really baseline T cells. Those are the male patients who had very low T cell activation went on to develop worse disease compared to those that had elevated levels. Again, there is no difference in the female. And that holds true for activated CD8 T cells or terminally differentiated CD8s or interferon gamma secretion from CD8 T cells. Conversely, female patients that went on to develop worse disease had elevated level of inflammatory cytokines such as TRAIL, CCL5, and IL-15. And this is also kind of a striking finding where when we plot the level of activated T cells, CD8 T cells and interferon gamma positive CD8 T cells across different age group in male and female, what we see is that men, as they age, they lose their ability to stimulate T cells or secrete interferon gamma from T cells. Whereas in the female group, there was no difference, or if anything, there's elevated levels of interferon gamma as women aged. So this told us that women age better with respect to the immune response, at least the T cell immune response to this particular viral infection. So here I conclude that men develop more inflammatory signatures, whereas they have impaired T cells compared to women at baseline. And men who developed very low T cell activation also ended up having worse disease outcome, whereas women who had elevated pro-inflammatory cytokines did worse with disease. And finally, as I said, women age better with respect to T cell immunity than men. So I want to conclude here by acknowledging the amazing team that I have the privilege of working with. I already mentioned Dr. Albert Koh and the IMPACT team. There are some wonderful clinicians that are treating the patients who are recruiting and really at the front line trying to treat these patients and make them better, like Dr. Charles de la Cruz and Shelly Farhadian, as well as Nathan Grubel, who is an amazing molecular epidemiologist who I had the pleasure of working with, looking at viral load as well as viral clade, where they come from. And Craig Weil and Aaron Ring are my colleagues in the department who are working very hard to look at the virology and antibody responses. And of course, my amazing lab members who are working around the clock on various aspects, not just looking at the biology of this infection, but also making tools and implementing testing. So I'm really, really fortunate to have this amazing people that I work with. And these are the authors that were involved in the 3 studies that I mentioned today. And of course, I thank the funding agencies for allowing us to be able to do this work. And finally, I just want to, again, emphasize my amazing lab members, but in particular, Karina Lucas, who's a Pew Fellow from Brazil. She joined my lab, relatively recently from Marcelo Bozza's lab. She has really spearheaded the longitudinal studies, and now she's on to something spectacular, looking at antibody response, which is yet to be published, but we are working on some really key issues around this immunity to coronavirus. So I want to end here and thank you for your attention.
Thank you, Akiko. Great talk. And thank people for this great audience. So a lot of people. We have some questions. First of all, I have a question. You recently demonstrated that SARS-CoV-2 reaches central nervous systems. And do you believe there is any difference between male and female for this susceptibility to the neuronal disease?
Yeah, no, that's a really great question. We only had 3 patients in that study, so we obviously cannot compare the sex difference, but we would love to expand this analysis to larger number to be able to see whether there's any sex difference there.
Okay, thanks. Mm-hmm. So let's see some questions. One from Ricardo Gazzinelli, the president of the Brazilian Society of Immunology. So many of the biomarkers of severe disease are common to various infectious diseases that cause systemic inflammation. Any immune biomarker or combination that you would consider COVID-19 specific?
Yeah, so I don't think there's anything extremely unique about those biomarkers. Potentially, it's something like a combination, as you suggest, might predict better the biomarker. We actually haven't done analysis using multiple factors, and that's a really good idea. Actually, we might do that. But yeah, I mean, some unusual things that has been reported, but not very frequent, are these eosinophils and IgE. This is really not that typical.
Mm-hmm. It is actually, I know. Mm-hmm. Another question from Wallace Pitanga. Do you suggest any explanation on why there is a significant elevation of IL-18 while this does not occur with IL-1 beta, since both cytokines mm-hmm. are produced through inflammasomes?
Right. No, that's another very interesting question. We certainly do see elevated IL-1 beta as well as IL-1 alpha, but it's not as dramatic as IL-18. IL-18 appears to be the most sensitive, sort of differentially sort of measured factor of all the inflammasome cytokines. We don't know the reason for this. It's possible that it's just a trivial, fact that we can measure more sensitively from the plasma. I know that some methods are much less capable of picking up the IL-1 family members. So it may be just a matter of technique, or it could really be biology that, somehow IL-18 producing cells are more stimulated compared to the IL-1 producing cells. So yeah, we don't know the answer to that yet.
Or some protease can degrade, I don't know,
mm-hmm. IL-1. Yeah, exactly, it's possible. Mm-hmm.
Yeah. So Fernanda Vargas Castanheira asked, so first, good talk. Have you looked at which cells in the mouse start to express ACE2 after ACE2 transduction, including immune cells?
That's right. So we are now looking at cell types that are transduced and are expressing and are capable of becoming infected. And because it's a AAV vector, it's not going to be cell type specific. And our promoter is also ubiquitous. So it's likely going to be transducing immune cells as well as epithelial and potentially endothelial cells. So we're really looking into that now, what cell types are transduced.
And Gustavo Amarante Mendes, hi Akiko, thank you so much for sharing your excellent thoughts and data with us. I have a couple of questions for you. Very intriguing data related with difference in male and female response to SARS-CoV-2. Do you believe this is related to hormones or to the way of life, including nutritional, exercise, etc.?
Yeah, so right now we don't know what the real answer is. We are measuring sex hormones from these particular patients to see if that can be the key factor driving the difference. But many of these people are postmenopausal and, ages of 80s and 90s. And so, we're not sure exactly what the contribution of the sex hormones in this case would be. Of course, some of them might be on replacement therapy and, so that's why we're actually measuring the blood levels of these hormones. And of course, the sort of more, behavioral aspect of sex differences might also play a role. We haven't asked them what they eat and how much they exercise, but that's also maybe a contributing factor.
Let's do yoga. Yeah. How does, the second one, how does the type 1 interferon signature related to the increase in netosis net formation observed in some patients with severe disease and to platelet activation and coagulation profile?
Yeah, so we don't know with this particular case, but when we studied the influenza virus infection in the mouse model, what we saw is an inverse relationship between interferon levels and netosis. So interferons, as you know, can inhibit transcription and activation of inflammasome factors, which is necessary for netosis. So it may be a reciprocal relationship. And even though when we say elevated levels of interferon, it probably is not enough to control the virus, and yet enough to allow the inflammasomes to become activated. So that's sort of a speculation answer, but I think there is a reciprocal relationship there.
Nice. Another question from Julio Schafstein. Considering that IgE levels seem to be upregulated in patients with worse clinical outcome, do you know if mast cells progenitors are being recruited to the alveolar septum in severe cases?
Yeah, so one thing we don't have any access to is the respiratory samples from these patients. So that would be a great thing to look at, what's happening locally, but we only have measurement from the blood. And and we cannot measure really mast cells in the blood. So yeah, that remains a mystery right now.
Okay. Dario Zamboni, thanks for that excellent talk, Kiko. You nicely show IL-18 inflammasome as well as type 1 interferon as biomarker for mortality. Type 1 interferon upregulates many processes, including autophagy, which negatively regulates inflammasome activation. So one predicts higher type 1 interferon and lower inflammasome activation. Is there a positive correlation between type 1 interferon and IL-18? Or some patient gets worse because of the type 1 interferon, while other get worse because of the inflammasome? Yes.
Yeah, that's an excellent question. Yeah, so I was just referring to that, but I think there might be a reciprocal relationship. But again, like the interferon levels that we're measuring is, even though it's elevated above the healthy control, it's probably very low levels. And so when we compare data from experimental human infection of seasonal influenza virus, interferon levels go sky high within 2 days of infection. And then it completely tapers down. Whereas what we're seeing, of course, we don't know what happened in the first 2 days of infection with SARS-CoV-2, but what we're seeing is that, days after infection, we're seeing still the smoldering levels of interferon in the COVID patients, indicating that, well, again, like completely suggestive, but the early phase interferon didn't happen. And therefore, there's this lingering viral replication driving low levels of interferon. However, I think that interferon isn't sufficient to control the inflammasomes. So I think, you just have this like negative effect of interferon contributing to activation of netosis, as well as inflammasome, as well as the recruitment of leukocytes that we showed in the animal model.
So many questions. Let me select one of them. From Hirogoto. How do you relate the cytokine profile suggesting triggering of inflammation and the finding in necropsy of patients where the inflammation is very scarce?
Oh, necropsy of patients. You mean the respiratory tract, or?
I don't know what she means, but maybe could be.
Yeah.
So
So I started the talk by saying there are so many different heterogeneous diseases associated with this. And let's say if the cause of death or severe disease has nothing to do with respiratory tract, right? It's possible that some people are suffering from more the neurological symptoms or cardiovascular symptom. It's also known to affect the heart even directly. So, the fact that we might not find the inflammatory signatures within the lung may not indicate that there isn't inflammation going on. It may be going on in different organs. So that's something, I mean, we've seen autopsy reports of this virus infecting pretty much every epithelial cells, right, in the body, intestine, kidney. So it may be that inflammation in different organs may be contributing to disease severity.
Okay. So 2 questions, one from Rafael Laroca and another one from Cândido Ferraz, related to some potential treatment. Rafael asked, did you try blocking IL-18? And Cândido, what would you think there would be benefit in type 1 interferon therapy in COVID-19?
Yeah, so I know that there are trials ongoing for IL-18 blocking antibodies or antagonists, but I haven't seen results of those types of trials yet. We don't know what happens. I mean, obviously that's the number one mortality factor. So blocking it might be beneficial, but we don't know. With respect to interferon therapy, I absolutely want to emphasize that, if you give interferon early, you may have a positive impact to block this infection. So I don't mean to paint interferon as the bad factor altogether. It's just that the endogenous interferon is likely not enough to control the virus, but is enough to induce inflammation. But endogenous interferon, as I mentioned, is probably delayed and too low. So if you were to give recombinant interferon therapy within the first few days of exposure, it may actually work much better than the endogenous interferon. And there are many trials ongoing right now, and some of them really do show that effect. So I do have hope for early interferon treatment.
Nice. Hope.
Yeah.
One question from José Carlos Filho, from the board of the Brazilian Society of Immunology. Thank you for the very nice talk. Regarding the longitudinal analysis of patients, what are the potential preconditions or factors that drive patients to the expression of tissue repair of gene signature?
Right. So the tissue repair gene signature was the one that was associated with cluster 1 patients who recovered from this disease. And so what are the sources? We don't know. We can only measure from the blood what we see. But we suspect that there are these tissue repair factors that are secreted locally in the infected tissue that allow for, let's say, repair of the epithelium, endothelium, fibroblasts, as well as IL-7, which indicates potentially lymphocytes could also be active, proliferating in these settings. So yeah, it was really gratifying to see that signature come up. Obviously, it's a AI algorithm that we had nothing to do with, but those are the features that we saw.
Nice. Montilo Russo, nice talk. What is the mechanism that control or allow virus replication? Do you know?
Oh, what is the factor that allow the viral replication?
Yeah, or control or
control. Yeah, control or, well, very good question. We don't know. I mean, normally as an immunologist, one should say, oh, it's the antibody or it's the T cells, but we don't have any evidence that it's either of those things yet, which is kind of frustrating, right? Because this many months into the pandemic, we still don't know what controls the virus. Yeah, very interesting question. We don't know. But all we know is that the consequence of this virus replication, but we don't know what controls it. And it does not relate to antibody levels. So in fact, the severe patients tend to have higher levels of antibody earlier than the moderate patients. And that's been reported by others. So I feel like it's not just the level of anti-spike antibody, but it's something else.
Thanks. One question about the mouse model from Juliana Maricato. Congratulations again. My question, is there any mouse model to evaluate SARS-CoV-2 infection tropism in the central nervous system?
Yes, in fact, we have the bioRxiv article where we transduce the brain cells using AAV. And that model works very well. You can infect so many brain cells with SARS-CoV-2 after introducing it into the brain. There's also the K18 animal model, which has ACE2 expressed under the K18, keratin 18 promoter. And that's also on in the brain. And you can infect cells of the brain using brain injection of SARS-CoV, as well as potentially after the intranasal infection. So there are models where we can study neuronal infection. And in fact, we did analysis of the entire brain infected after this AAV transduction. And it's like, it lights up the entire brain. So it's very, very permissive to infection.
Thanks.
Mm-hmm.
One question from Cristina Ribeiro, also from the SBI Board of Directors. Do the different phases of ovulatory cycle or menopause impact in the immune response to the virus in women, since it seems to be a gender-biased disease? The response to therapies such as corticoid is different among them.
Yeah, so again, it's a very intriguing possibility, right? That the sex hormones might be controlling this immune response difference that we see. And people actually have already trials ongoing to treat, men with estrogen, for instance, to see if that would help them. I don't know the outcome of those trials, but that should tell us something about whether or not sex hormones can impact the disease. And as I mentioned, many of our patients were postmenopausal. So again, like unless they're on hormone replacement therapy, sex hormones is likely not the major factor, but we are still measuring them, yeah.
One interesting question from Maria Sato. TLR7 and ACE2 are encoded by a gene on X chromosome on X chromosome. Do you think that male COVID-19 susceptibility is related to this?
Yeah, so it may very well be X chromosome linked. So the TLR7, it's been reported that TLR7 deficient men can develop very severe disease. This was reported in JAMA. And so it's clearly an important innate sensor that women have two genomic copies of. Even though the X inactivation is repressing the other set, but it's not complete. So it's possible that the elevated level of TLR7 is resulting in this better T cell immunity and eventually control of disease. We don't know. We're actually testing in animal models to see what happens.
We have many questions yet. Still have many questions. I will select. So one from Aurunomizu. Are you looking for cells producing IL-10? Do you considering that it plays some role?
Yeah, so IL-10 was also like the third top mortality associated factor. It doesn't mean that IL-10 itself is contributing to mortality. I think it's like a body's attempt to regulate the inflammatory response, and that's why it's up there. But yeah, we don't know what cell types are secreting, potentially macrophages, but we don't know the source of IL-10. It's likely important to repress some of these hyper-inflammatory signals, but yeah, we don't know the source.
Another one from Magnus Gidlung. Excellent presentation. My question concerns the role of MHC molecules in the response to COVID-19.
Yeah. Yeah, very interesting question. Yeah, obviously our study wasn't powered enough to look at the HLA types because we only had, 100 patients or so. But maybe in larger epidemiological studies might reveal HLA association. Lots of diseases obviously have the HLA association for both protection and disease exacerbation. One of the things that I speculated earlier during this pandemic, when I was asked why Japanese why Japan is able to control the pandemic so well, I speculated potentially something genetic like HLA, but we don't know. There's also the BCG theory and many other theories out there. So yeah, it's something to look out for.
Okay. And Luis Rodriguez Jr. asked, do you think that pulmonary TRM, T-resident memory cells, have an influence on the severity of COVID-19?
Yeah, so there was a very nice perspective published by Shane Crotty and others recently in Nature Review Immunology that speculates the role of tissue-resident memory cells. And according to that, so what we know right now is that SARS-CoV unexposed individuals, up to 50% have pre-existing CD4 T cell immunity that cross-reacts with SARS-CoV-2. And whether that cross-reactive T cell is protective or harmful or completely neutral, we don't know. One of the speculations is that if the CD4s are in the lung or in the nasal cavity, they might be able to re-stimulate the memory response quickly to be able to control the virus or at least control disease from the virus. And so my suspicion would be that TRMs would contribute somewhat to the control of the virus locally. That would obviously protect the rest of the body from becoming infected. We have no evidence of that yet. It's a speculation.
Okay. Ricardo Gazzinelli, please give my congratulations for the great talk and thank her for accepting our invitation. Again.
My pleasure. Thank you.
One more from Gustavo. A more general question related to the actual state of disease in the world and the idea of herd immunity. Is it time to let the disease spread among the young people to try to reach herd immunity, or should we be patient and a bit more and wait for the vaccine to be released to public?
Yes.
Please ask this question.
Yeah, I get asked that question a lot from reporters because it's really being talked about in many different countries. But I stand firmly that we need to be patient to wait for the vaccines, because even the young people who are infected have many thousands of people have this long hauler disease, which is debilitating them for months and months. It's a horrible state that they have to go through. And we also don't have any treatment for the long hauler disease yet. So I would not take this virus lightly. Even young people, children, anyone is susceptible to severe disease from this infection. So herd immunity, and the fact that the naturally acquired immunity is not very robust. We're seeing waning of antibody as well as immunity to this virus within a year, with the reinfection cases coming up. So I cannot advise herd immunity acquired by natural infection with this virus.
Yes, and I have another question regarding the potential. Do you believe that antibody-dependent enhancement could be a problem with the vaccination against the SARS-CoV-2?
Yeah, so it's not an immediate concern, because we haven't seen any evidence of ADE in the animal model, as well as phase 1. Well, phase 1 will not show that, but it hasn't really revealed itself with any of the vaccine candidates yet. But it's something that we really need to keep an eye on for the phase 3 trial, to see if any of the exposed people are getting worse disease, and if so, what is going on. I think we cannot let our guards down yet and dismiss the possibility of ADE, especially antibody-dependent enhancement of disease, because that has happened with the inactivated SARS virus vaccine before. So we do have to keep an eye on that.
Okay, thanks. Thank you, Akiko, again for your patience and for answering many questions, and for your great talk. And especially thank you for the huge contribution to the understanding this immune response to SARS-CoV-2. It is so important for all of us in this moment.
Thank you very much, Karina, for moderating and asking great questions. And it is my pleasure to have been here. And if you have any questions, just email me, people who I didn't get to answer.
So thank you again.
Thank you.
Thank you, people, for the audience and for these questions. Thanks.
Okay. Bye-bye. Thank you.
Bye-bye. See you.
See you.
Mm-hmm.